What happens when you vaccinate somebody and then expose them over and over again to the same virus? That's really the question of a challenge study. And normally it's used in the context of science or in research to try and identify if there are going to be issues in the future. And so sometimes they would do an animal challenge study. This was done in 2000 and about 11, 12, when they injected the animals with different types of SARS-CoV vaccines. So this was the original virus from 2003. And then what they did is that they then exposed them to the virus to see what would happen. And in 2012, they found that all of the animals developed a hypersensitivity response on exposure to the virus. So that was quite important. And at the time, they had said we need to use caution when we are using this kind of vaccine in the context of human studies. Now, some may argue that we're using different types of vaccines, and they're right. Certainly, the mRNA vaccines has the advantage from the perspective of this hypersensitivity response that they produce IgG4. That's the tolerant antibody. And so, quite remarkably, they may be less likely to have immune pathology on re exposure to the virus, but that's just in the lungs. One of the things that I've been highlighting for some time is this concept of the COVID storm. And the COVID storm effectively means that we have a situation where, on re exposure to the virus, when it gets circulating in the bloodstream, this can then cause other kinds of immune pathology in the body. So, this is the other side of the picture. But in terms of lung involvement, this is an important question for which we don't have an answer. I just know from previous studies this was a problem. The question is it going to be a problem going forward with high circulation of virus as we're seeing in highly vaccinated regions? Because that's the category or that's the description of a challenge study. Now, in terms of the unvaccinated cohort, they may be thinking to themselves, well, I'm not concerned. This is not relevant to me. I would advise caution when you're thinking like that because it's not so straightforward. And this raises one of the questions that had been around for the longest while with regards to shedding. And it was debunked when they were talking about viral shedding. But what is scientifically logical is lipid nanoparticle or exosomal shedding. I'll explain what that is in just a second. This here represents an exosome. And an exosome is produced normally by cells when they want to pass information around. Sometimes it can be cytokines, DNA, RNA, different things. And in this case here, we have an extracellular vesicle that could have multiple things in it, including mRNA and potentially spike protein. This would be therefore in somebody, say, who had been vaccinated and they have circulating extracellular vesicles. In theory, when we think about that, we have to realize that those extracellular vesicles can then circulate in the bloodstream and also end up in plasma. And if it can get up in plasma, it can therefore get in saliva. And so theoretically, you could have a situation that occurs simply like this. This is where the exosome that was in the saliva now, not just the bloodstream, coughing, sneezing, it spread to somebody else who is this in the vicinity. And if they inhale that and they're exposed to the particles in here, and they have previously been primed, they could get hypersensitivity responses similar to what could happen just with pollen. So it's not something that we can necessarily discard completely, but it is something that if it is happening, you would therefore oftentimes have associated symptoms where on exposure, if you're in contact with people like that, you would have unusual symptoms. You could have headaches, you could have coughing, sneezing, that kind of thing could occur. These are the kinds of things that I'm trying to see if I can figure out. And this is why I want you to make sure you join me. Look in the link below for sessions 2 of McMillan and members. This is where we look at this kind of thing. There is, I'll be collecting information from people with regards to their particular situation, where we can then try and create a scenario that would help people to be able to understand this better. But these are important questions. What really happens? How does it manifest? And in truth, what I expect to happen is that we're going to have a surge in respiratory diseases as the virus continues to circulate. And you have this concept of exosomal hypersensitivity responses. All these things coming together could be quite a serious challenge for us to face in the future. But the sad part is that there is no actual research going on about this. This should actually be studied. Normally, what would happen in a challenge study is that you would expose the participants after they'd been vaccinated, and then you want to confirm by looking at the immune pathology in the lungs. This could be like biopsy or bronchial alveolar lavage to see exactly what is happening to the cells. That's what you would want to do to try and understand it. At this point, it seems nobody really cares. The pandemic either has passed or the vaccines are out there and there is no longer a problem. We need to be far more aware of what is going on and recognize where the limitations in research actually are. There are some big challenges ahead. And I am certainly concerned about how highly there is circulation of virus in terms of highly vaccinated populations. Some people say, well, why are you only concerned about them? Well, the reality is that in a low vaccinated region where they have mucosal immunity, it will stop the virus before it can efficiently spread. In a situation where you have poor mucosal immunity, like what is happening in highly vaccinated regions, they'll get infected over and over. And therefore, they put everyone at risk, not just the vaccinated, but the unvaccinated who will be continually exposed to different viruses. This is the problem. And it's important to clarify. People may sometimes think that because they haven't had COVID infection in their minds, they haven't had overt symptoms, they are fine. You have to remember, it's not that the virus stops at your mouth. It gets into the airway. It may try and infect the epithelial cells, the lining cells, but because you have a strong mucosal response before it can get very far, so it does cause an infection, but the infection is limited and it's stopped by your immune system. And someone may think to themselves, well, that's great. I don't have a problem. The issue is that when you think about pollen exposure, you just need to be exposed to it for it to cause hypersensitivity responses. That's the bit that I'm concerned about because if the spike protein can trigger hypersensitivity responses, then the unvaccinated are just, if not potentially, at higher risk for that kind of issue than even their vaccinated cohorts. We need to understand it. We need to look out for it. And critically, we need to find ways to mitigate it if this is happening. So, again, I encourage you that I'm very happy to discuss this kind of thing to try and get some insight from your perspective. Join me with regards to Macmillan and members. Our session is coming up. And guess what? We have also started our process with regards to looking at disease X. We are on the brink of being able to put out our book that we've been working on for a long time. We're just in the final stages now. Please join our Kickstarter program. We are already over 50% of the target that we have pledged, and we would look forward to your support. So please join us in this process. The more information that you have, the better we will all be prepared for the future. So let's continue to explore the science, ask the hard question, reflect on the pathology that can happen and affect everyone around us. So let's not make sure that we are always on top of the things that we are facing. So have a good evening until we speak again. Thank you.